Hello students, today in this lecture series, I'm discussing with you second BSc, third semester biotechnology, NEP practical BCU, paper biomolecules, practical scheme. Okay, before going through, let us go through the practical question paper, subject biomolecules, code DCBT-P301, third semester BSc practical examination, 2023. Time 4 hours, maximum marks 25. Question number 1, estimate the amount of protein present in the sample A by Lowry's method or Biuret method. And this carries 7 marks. Or R, estimate the amount of reducing sugar present in the sample A by DNS method. So in question number 1, you have option Lowry's method or Biuret method or DNS method. Here you need to write the principle in short and procedure in the form of tabular column, plot the graph and write the result. Question number 2, determine the activity of amylase in the given sample B. Write the principle and procedure in detail. And this carries 8 marks. R, extraction and estimation of protein from soaked or sprouted green gram by salting out method. Okay, this carries 8 marks. Then question number 3, write the principle and procedure involved in any one of the following. Here, this question carries 4 marks. You may get estimation of amino acids by ninhydrin method or separation of plant pigments by paper chromatography. So ninhydrin or paper chromatography. In these two, any one you need to write in the examination. Any one will be asked in the exam. Okay, and this carries 4 marks. Then question number 4, describe the principle involved in any two of the following. And this carries 3 marks. From this A, you may get TLC or page. First, write the full form of this and comment on this particular TLC. No comment. They have given here, describe the principle. So, to write the principle behind this TLC or maybe a page. And even preparation of buffers. Then saponification, add the number of lipids. Or you can also comment on this particular TLC page, preparation of buffer, how are you going to prepare buffer or definition of the buffer and what are the different types of buffer like that. Then saponification, add the number of lipids. Okay. Then 5th one is practical record. This carries 3 marks. Now let us go through the brief information about the answers of question number 1, 2, 3, and 4. Question number 1, you are writing here for Lowry's or Biuret or maybe a DNS method. Okay, so let us go to the first one, Lowry's method. To estimate the concentration of proteins by Lowry method from the given sample. Okay, here you need to write the principle in brief, then write the tabular column. Okay, properly. First one will be the blank and 7th one will be your unknown. Okay, and concentration is supposed to keep in your mind here, and that is in the microgram. Okay, concentration of protein, you need to write here 40, 80, 120, 160, 200. Okay, then after adding FC, okay, incubate at room temperature, the dark for 30 minutes, then take the OD value at 700 nanometer or 660 nanometer. After plotting the graph, then write the result without fail. The concentration of protein in the given sample dash amount of microgram per ml. Okay, this is about the Lowry's method. So when you want to go through the graph sheet, this is the graph. Okay, first write the title of the experiment. Don't write below, top. You need to write the title of the experiments, and once you receive graph sheet, kindly write the graph sheet number in the register. Okay, then you write the scale, how much are you going to take for x-axis, then y-axis. X-axis 1 unit is equal to dash, y-axis 1 unit is equal to dash. And this is the x-axis. This x-axis, first write concentration of protein in microgram. Then y-axis, OD at 660 nanometer or 700 nanometer. So here 1 centimeter, we have taken 40, 80. Okay, like that concentration is taken here. This is the this concentration you are going to plot here. Okay, and this is the OD on y-axis. This is all about the graph sheet. So compulsory, whenever you're going to plot the graph, title of the graph and scale, how much unit on x-axis, how much unit on y-axis, you need to write. Then write what is on x-axis here, then y-axis, then standard straight line you plot, and based on the unknown OD, okay, with respect to this straight line, plot the graph. And you need to plot the unknown graph always in dotted line, and wherever this dotted line touches, that much amount of microgram of protein present in the given sample. So this is question number 1, Lowry's method. Next, our biurate method. Here to estimate the concentration of protein in the given sample, biurate method. So this is the principle behind the experiment. Procedure in the tabular column. Compulsory, you need to write the procedure in the tabular column only, not in a sentence flow chart. Okay. Then result you need to write the given unknown sample found to be dash amount of microgram protein per ml. And this is the graph here. Okay. Always write the title of the experiment on top of the graph. Write the scale. X-axis 1 unit is equal to how much? Y-axis 1 unit is equal to how much? Okay. Here is written x-axis concentration of protein microgram. Here 1 centimeter, we have taken 1,000, 200,000, 3,000, 4,000, 5,000 like that. Here concentration of OD at 560 nanometer. Okay. Is this clear? Then plot the graph 560 or maybe 540 nanometer. And this is the standard straight line you need to plot. Then unknown in dotted line you need to plot, and wherever this dotted line touches here, this will be the your unknown concentration of the protein in that given sample. Next one here, estimate the amount of reducing sugar present in the given sample A by DNS method. Right, first you need to write the aim, then write the principle, okay, and write the reaction. Then this is the tabular column. First, volume of maltose solution we have taken, where this maltose is prepared by using standard volumetric flask. Then distilled water you need to make up. Then concentration of maltose goes like this, like 200 microgram, 400 microgram, 600, 800 and 1000 microgram. Then volume of DNS in all the test tube 1 ml. This is last one is the unknown. Okay. And here you need to incubate. Then OD at 540 nanometer. Write the result based on the concentration of the unknown. The amount of reducing sugar in the given sample is dash microgram per ml. Okay. Graph is very important. Focus on the graph. Always write the experiment title on top. Then write the scale. X-axis how much? 1 unit. Y-axis how much? 1 unit. Then here also write X-axis concentration of reducing sugar in microgram. Then here OD at 540 nanometer. Then whatever the OD you got, based on that, you need to plot the straight line. Then unknown, you need to plot here. Wherever this unknown touches, no, that will be your concentration of the maltose which are present in that particular given sample. So this is 600 microgram unknown sample. So always the unknown must be plotted in dotted lines. This is about DNS method. Then question number 2, determine the activity of amylase in the given sample B. Here you need to write the principle and procedure in detail, and this carries 8 marks. For example, if you get this particular salivary amylase enzyme by DNS method, question number 2, and in the question number 1 also you get DNS method, means one standard can be used for both the experiment. Okay, and this is the principle you need to write. Okay, given principle in brief you write. Okay, please you can take screenshot and you can study this one. This is the principle, and they have told principle procedure in detail. So this principle and this procedure need to write. So procedure, first preparation of the standard graph is given here. You are going to weigh 100 mg of maltose dissolved in 100 ml of distilled water. So all this will be prepared in the standard volumetric flask in the examination hall and kept. Not necessary to worry, you need to take only the chemicals. So you need to keep this in mind. So how much concentration we have prepared here? 1000 microgram per ml. Okay. So this is for the salivary amylase activity. You can pause this video or you can take screenshot for the study purpose or writing purpose. Okay. Then we need to go for the next one, that is enzymatic reaction and determination of the enzyme activity. Okay. So here you need to take the 0.5 ml of properly diluted crude enzyme. Okay. Then incubate in 0.5 ml of starch for 15 minutes. Then your activity of the enzyme calculation you need to apply, and you need to find out that how much amount of the activity has been done. So this is the observation. This tabular column you need to write, okay, and the enzyme activity tabular column also you need to write. Here sometime the OD will be given. This OD you can keep in your mind, and to perform this test, and the blank one you need to perform. Okay, then you can plot the graph. Estimation of alpha amylase activity by the DNS method. Standard will be given. You need to check the unknown sample. As usual, whenever you plotting the graph, first write the scale, then title of the experiment, then what is x-axis, y-axis, all those things you need to keep in your mind. Then write the unknown once you finish your graph. And this is the calculation for the enzyme activity. 1 ml saliva is diluted to 50 ml. From this, 0.5 ml was taken for determination. Then you need to apply this particular formula to obtain dash amount of micromoles per minute. The result is very, very important. You must write the result. Result carries 1 mark. Okay. So let me go through this one, enzyme activity. Please take screenshot and study this one. Determine the activity of amylase and the given sample B. Write the principle and procedure in detail. So this principle and this procedure, preparation of the standard graph, and this enzymatic reaction and determination of the enzyme activity, up to here you need to write in detail. Then you need to plot the graph, you need to write this tabular column, okay, to find out the enzyme activity. Then you need to write this particular table. Okay, so table follows like this, then graph. Okay, then you also focus on this particular calculation part, then write the result. Then in the question number 2, you have another option, that is extraction and estimation of protein from soaked or sprouted green gram by salting out method. This carries 8 marks. So here this is the same question number 2. You may get enzyme amylase activity, or you may get the salting out method. Okay, here this is the aim of the experiment, then principle and procedure. In this experiment, there are 2 concepts. One is extraction, other one is the estimation. First, you are going to extract the protein from the green gram, okay, sprouted green gram. Then in that green gram, how much amount of protein is there, you are going to estimate. So there are 2 concepts are there. First, in procedure, you are going to write the preparation of plant extract, that is extraction methodology. Okay, you are going to weigh and you are going to grind. All those things you are going to do here. That is extraction methodology. Then step 2 is estimation. So here you are going to write the procedure how to estimate the proteins. So in salting out method, we are using the biorate method. Okay, in the enzyme activity method, we are using DNS method. Whereas in salting out method, we are using biorate method. So this is the procedure to perform the biorate method. So whatever you have written in the studied that biorate method, no, the same biorate method follows here for the salting out method. This biorate method you need to keep in mind. This is the procedure form of flowchart, okay, in sentence form, or you can also write this in the form of tabular column. Okay, here you can see the biorate method. So I hope you're able to understand. Plant from the green gram protein extracted. To that extracted protein will be taken in the 7th test tube. That is nothing but the unknown. All 6 will be the standard you are going to get. You are going to weigh the BSA. BSA full form, bovine serum albumin. Okay, once you get the standard, on top and to the standard, you are going to plot this unknown constant, and you are going to plot the unknown OD, and then you will get the some amount of concentration microgram of protein. And the salting out we are performing here for 45% of the green gram. Okay. Then you need to plot the graph like this for the estimation of protein from soaked green gram by salting out method. First, write the title of the experiment on top, then scale you write what is x-axis and how 1 unit, 1 y-axis, how much amount. Okay, then here also x-axis you write, here also y-axis, and this is the standard straight line, and plot the unknown graph by in dotted lines. And wherever this dotted line touches, this will be the concentration of the proteins present in that particular sample. So question number 1 and 2, you are performing the experiments. Okay. Then question number 3, only writing part. Question number 4, only writing part. Here, question number 3 is write the principle and procedure involved in any one of the following. Okay. You may get here estimation of amino acids by Nin-Hydrin method, or you may get the ascending paper chromatography. Okay. In these two, any one you are going to get Nin-Hydrin method or maybe a paper chromatography. So here they have already mentioned, write the principle and procedure. So you need to write the principle behind this Nin-Hydrin method. What is this Nin-Hydrin method? And what happens when the Nin-Hydrin reacts with the amino acid, that you need to write, and remains purple color you need to write, and OD also you need to write. So this is the principle, this is the reaction, and this is the procedure. Okay, as per they was write the principle and procedure, you need to write the principle, and this is the procedure, and this carries 4 marks. So this is all about Nin-Hydrin method. So in the exam, you may get Nin-Hydrin method, or you may get this particular paper chromatography method. So in the previous table, I have showed this is the procedure form of sentence form. Same procedure can also be written in the tabular column. Okay, better go through the procedure form of this sentence form or maybe a flow chart. This is enough for the examination point of view. When you are performing the Nin-Hydrin method, this table and the result is important. Now let us go through separation of plant pigments by ascending paper chromatography. You can take screenshot and you can keep this ready. So here in separation again, they have asked the principle and procedure. So here you can see the principle and this is the procedure. This carries 4 marks. Okay. Take screenshot or pause video and kindly go to this one. Study this one by yourself. So this is all about question number 3, Nin-Hydrin method and paper chromatography. Okay, so this is the result of the performance of paper chromatography. If you want, you can also write this particular plots like this. First pencil line, then solvent front, okay, then second line, then pigment, third and fourth. So you can write a lot of pigment showing in paper chromatography like that. And the fourth one is describe the principle involved in any two of the following. This carries just 3 marks. Okay, you may get TLC or page. From this A, you are going to get any one. From this B, you are going to get any one. Here TLC, first write the full form, then focus on this. What is TLC? Then applications you can tell. We can write the principle. In general also you can write. Don't worry that only they have asked principle like that. TLC, we can write. Then page. You can write first write the full form and then focus on its principle of the page. Then preparation of buffers. What is buffer? What are the different types of buffer applications you can write? Okay, then saponification, then iodine number of lipids. Any two you are going to write. Each answer carries 1.5 marks. So minimum 4 points is more than enough. So here first one, principle of thin layer chromatography I have given here. Please just go through this one. Okay, study this, pause the video and go through this particular TLC. In general, I have given. Okay, you can write extra information also, not a problem. See to that to write minimum 5 points. Okay, and the next one here, principle of native page. Okay, so page this much if you write, that is more than enough. Write the full form and write further information about the page principle and the purpose, everything. Okay. The next one is principle of preparation of buffers. Okay, you can in general also you can write what is buffer, what are the different types of buffer and formula, all that things you can also mention here. And also compulsory need to mention this Henderson-Hasselbalch equation without fail for this particular preparation of buffers. Okay, as I told earlier that take screenshot or pause this video and go through this preparation of buffers. Next, coming to the principle of saponification value of lipids. As you already studied this one in the theory class, you can write that one and you can score better marks. Kindly focus on this particular principle of saponification value of lipids. Can focus on definition of the saponification and how the saponification will be done. Application, as in the form of applications also, you can write this related with this particular saponification. Now, coming to the end, and the last one is principle of odd number of lipids. Okay, please go to this one. Whatever information is given here, okay, that much if you write more than enough. If you know additional any information, well and good, you can write that also. First, define what is this RD number and write about the purpose of this RD number of lipids. Okay, so I hope you understood today's concept about the third semester practical scheme. Let me go through one by one here. The first one is this question pattern we studied, right? Lowry's method, pyruvate method, DNS method. Then activity of amylase, then salting out method, ninhydrin method, paper chromatography method, TLC, page, preparation of buffers, saponification, and the number of lipids. All these things you need to focus on and refer this particular video. That is more than enough. For Lowry's method, whatever is given here, principle, procedure from tabular column, graph, result is must. Okay. Next one is the pyruvate method, principle, the tabular column, graph, and this result is very, very important. Then for DNS method also, same thing, principle, procedure in the form of tabular column, then graph, and then result is very, very important. Okay, then question number 2, amylase activity. They've asked in here in the question only write the principle and procedure in detail. So write the entire principle, then procedure, preparation of the standard graph, then enzymatic reaction, determination of the enzyme activity, and this formula. Then these two tabular column. Okay, in the form these two tabular column itself is a procedure, or you may also call observation as you are recording here the OD. Okay, this is very important. Then write the result, then plot the graph and put the calculation. Okay, then soaked sprouted green gram, principle, 2 steps procedure, preparation of plant extract, estimation of protein by pyruvate method, and the respective tabular column, and write the result, plot the graph. Next, ninhydrin method and the suppression of plant pigments by paper chromatography. Only you need to write the principle and procedure. So this is the principle and here is the procedure. Okay, and this carries 4 marks. If possible, keep in your mind this reaction. Otherwise, this principle in sentence form, that is more than enough. Then this ninhydrin tabular column is not necessary as you're not performing, only writing part. Then this is attending paper chromatography, principle and the respective formula, then procedure. Okay. Then here you can see about the TLC. Okay. Then page. Then this is preparation of buffers. Then saponification value of lipids. Then principle of RD number of lipids. Okay, this is all about your third semester paper, biomolecules practical scheme and what all you need to write in the examination. I hope you're happy. Best of luck. Stay blessed. You be the gift to the world. Thank you everyone for your patient listening. Thank you all.